What if humanity's survival didn't just depend on adapting to new environments, but on designing life itself to thrive where no life has ever existed? From the dusty plains of Mars to the distant, uncharted worlds of exoplanets, the idea of creating entirely new ecosystems tailored for survival might sound like something out of a sci-fi blockbuster. But this isn't fiction anymore, it's the cutting edge of science. Synthetic biology, a field where biology and engineering collide, is paving the way for humanity to not just explore space, but to truly inhabit it. In this video, we're diving into how this groundbreaking technology is enabling us to design organisms capable of transforming inhospitable worlds into livable habitats. From solving the problem of food and oxygen shortages to shielding us from cosmic radiation, synthetic biology holds the key to building sustainable life beyond Earth. So, how exactly are scientists rethinking what life can be in the pursuit of colonizing the stars? Let's find out. At its core, synthetic biology is like giving scientists the ultimate toolkit for life itself. It's a discipline that blends the precision of engineering with the complexity of biology, allowing researchers to design and build new biological systems or reprogram existing ones to perform specific tasks. Think of it like coding, but instead of writing software for computers, scientists are rewriting the genetic instructions that govern living organisms. The concept of synthetic biology traces its roots back decades, to when scientists first began decoding DNA and understanding how genetic information directs life. But in recent years, thanks to groundbreaking advancements, this field has undergone a revolutionary transformation. Two key innovations at the heart of this progress are CRISPR-Cas9 and gene circuits. CRISPR-Cas9, often described as molecular scissors, is a tool that enables scientists to edit DNA with unprecedented precision. Imagine being able to cut and paste sections of genetic code, much like editing a sentence in a document. With CRISPR, scientists can turn genes on or off, insert specific traits, or even remove harmful mutations. It's already been used to create disease-resistant crops, engineer bacteria to produce medicines, and even explore potential cures for genetic disorders in humans. But in the context of space exploration, CRISPR opens the door to customizing life forms that can survive and thrive in harsh, alien environments. Gene circuits, on the other hand, are like tiny biological computers. Just as electrical circuits use inputs and outputs to perform tasks, gene circuits are networks of genes that can be programmed to respond to specific signals. For example, a gene circuit could be designed to detect low oxygen levels and trigger the production of oxygen-generating enzymes. These kinds of biological programs allow us to build living systems that can respond dynamically to changing conditions, a critical capability for life beyond Earth. What makes synthetic biology so powerful is its versatility. Using these tools, scientists are rewriting the blueprint of organisms to fulfill specific purposes. Take algae, for instance. By engineering algae to produce biofuels or essential nutrients, researchers are creating sustainable solutions that could replace traditional energy sources and support life in isolated or resource-scarce environments. Similarly, crops can be modified to grow in extreme climates, whether that's the freezing cold of Mars or the acidic, rocky terrains of distant planets. These technologies are remarkable here on Earth, but they become absolutely essential in the vast emptiness of space. Imagine designing microbes that can extract valuable resources from Martian soil or plants that can grow with minimal water and sunlight while producing breathable oxygen. Synthetic biology is more than just the next big thing in science. It's the foundation for creating ecosystems that can help humans move beyond Earth and survive the challenges of deep space. But as exciting as these breakthroughs are, they also provoke some fascinating questions about the ethics, risks, and limitations of reshaping life itself. Who decides how we alter these genetic blueprints, and how do we ensure these technologies are used responsibly? These are questions we'll need to explore as we continue down this path. For now, what's clear is that synthetic biology has given us the tools to rethink what's possible. It's not just about adapting to the future, it's about building it from the ground up, one engineered organism at a time. And when it comes to space colonization, these innovations aren't just game-changing, they're survival-critical. Space is an unforgiving frontier. It's vast, cold, and void of nearly everything humans need to survive. There's no breathable air, no abundant water, no fertile soil, and certainly no grocery stores around the corner. Traditional approaches to space exploration have relied heavily on sending everything astronauts need from Earth, from food and oxygen to spare parts and fuel. But as exciting as the idea of living on Mars or traveling to distant exoplanets might be, the sheer cost and logistical challenges of transporting resources across the cosmos make this approach unsustainable in the long run. This is where synthetic biology becomes a game changer, offering solutions that traditional technologies simply can't. To understand why space needs synthetic biology, we have to consider the unique challenges of long-term space exploration. One of the most pressing issues is the lack of self-sustaining systems. On Earth, we're surrounded by ecosystems that recycle essential resources. Plants provide oxygen, microorganisms break down waste, and rain replenishes our water supplies. In space, none of these natural systems exist. Every breath of air, every sip of water, every bite of food must be carefully accounted for. The longer the mission, the harder it becomes to sustain life without constant resupplies. Traditional engineering can't fully replicate the complex cycles of life on Earth, but synthetic biology can. Imagine a spacecraft equipped with synthetic microorganisms engineered to recycle carbon dioxide into oxygen, much like plants do, but far more efficiently. Or picture a colony on Mars where crops are bioengineered to thrive in Martian soil, enriched by microbes tailored to extract nutrients and detoxify harmful compounds. These solutions don't just supplement existing systems, they redefine what's possible in terms of self-sufficiency. Synthetic biology enables us to create closed-loop life support systems, where waste is transformed into resources and every molecule is reused, drastically reducing dependence on Earth. Food production is another massive challenge. Current methods involve pre-packaged meals carefully rationed for months or even years, but this model becomes impractical for longer missions or larger crews. Instead, synthetic biology could enable us to grow customized, nutrient-rich food on site. For example, imagine crops engineered to grow faster, require less water, or even produce additional vitamins and proteins for enhanced nutrition. Or consider microbes that can ferment simple sugars to create everything from meat substitutes to dairy products, on board and on demand. These innovations mean astronauts wouldn't just survive, they'd thrive with access to fresh and varied diets, even millions of miles from Earth. Let's not forget materials. Building habitats, repairing equipment, and even manufacturing tools all require resources that are costly and bulky to transport. Synthetic biology opens up the possibility of producing these materials in situ. Picture bacteria engineered to produce bioplastics or even synthetic spider silk, a material stronger than steel but lightweight and versatile, using raw materials found on the Moon or Mars. Such breakthroughs could enable astronauts to print parts and construct shelters with minimal input from Earth, transforming the way we think about colonizing other planets. Perhaps one of the most intriguing applications of synthetic biology is its potential to protect humans from the harsh realities of space. Cosmic radiation, for instance, poses a serious threat to long-term missions beyond the Earth's magnetic field. Traditional shielding often relies on heavy materials that are impractical to launch into space, but synthetic biology could provide an elegant alternative. Microbes engineered to produce radiation-resistant compounds, or even human cells enhanced with borrowed genetic traits from extremophiles organisms that naturally survive high radiation levels. This would offer a layer of biological protection that's lightweight, scalable, and adaptive. The beauty of synthetic biology lies in its ability to transform challenges into opportunities. It doesn't just solve problems, it does so with elegance, efficiency, and adaptability, traits that traditional technologies struggle to replicate. When it comes to space exploration, we're not just trying to survive the challenges of a hostile environment. We're trying to build a life, a sustainable, thriving existence beyond our home planet. Synthetic biology is the toolkit that makes that dream possible. Now, let's take a closer look at one of the most ambitious and exciting applications of synthetic biology, designing organisms specifically for Mars. Mars is often seen as humanity's next big step, a planet that, while harsh and unwelcoming, has just enough potential to one day support life. But here's the thing, Mars isn't Earth. Its surface is icy cold, with temperatures plunging well below freezing. Its atmosphere is thin, composed mostly of carbon dioxide, and there's barely a trace of oxygen. Add to that the lack of liquid water, high levels of cosmic radiation, and the toxic chemicals in its soil, and it becomes clear why simply showing up and planting a flag isn't enough. If we're going to live on Mars, we'll need to radically transform its environment, or at least find ways to make its resources usable, and that's where synthetic biology comes in. Imagine microorganisms custom-built to survive Mars' extreme conditions. Scientists are already hard at work creating bioengineered microbes that could transform Martian soil into fertile ground for agriculture. Right now, Martian soil, or regolith, is hostile to most terrestrial plants. It contains perchlorates, toxic compounds that would kill most living things, and lacks the nutrients plants need to grow. But by introducing engineered microbes, we could start to terraform the soil, breaking down harmful compounds and enriching it with nitrogen, phosphorus, and other essential nutrients. Essentially, we'd be giving Mars a biological makeover, turning barren ground into something capable of sustaining crops. And these microbes wouldn't just stop at fixing the soil. Synthetic biology is also being used to develop organisms that can produce the essentials for human life, like oxygen. Some scientists are working with cyanobacteria, a type of microorganism that's incredibly good at photosynthesis. On Earth, cyanobacteria helped create the oxygen-rich atmosphere we breathe today, billions of years ago. On Mars, engineered cyanobacteria could perform a similar role, converting carbon dioxide from the Martian atmosphere into breathable oxygen. These microbes could be deployed in bioreactors or directly into Martian habitats, providing a steady supply of oxygen for humans while also laying the groundwork for larger-scale terraforming efforts. But it doesn't stop there. One of the most innovative ideas involves using synthetic biology to create biofuels on Mars. This is crucial because transporting fuel from Earth is both expensive and inefficient. Scientists are now exploring the possibility of engineering algae or bacteria that can convert Martian carbon dioxide, water from subsurface ice, and other raw materials into biofuels. These fuels could power everything from spacecraft to colony equipment, reducing the need for constant resupply missions from Earth. With the right organisms, we could potentially create a self-sustaining system where food, fuel, and oxygen are all produced locally, using the resources Mars already provides. Of course, designing life for Mars isn't just a matter of tweaking DNA and hoping for the best. it requires a deep understanding of how life operates in extreme environments. To that end, researchers often draw inspiration from extremophiles organisms on Earth that already thrive in conditions similar to Mars. For example, certain microbes found in Antarctica can survive in freezing temperatures, while others from deep sea vents tolerate crushing pressures and toxic chemicals. By studying these extremophiles and borrowing their genetic traits, scientists can build organisms that are not only tough enough for Mars but also tailored to perform specific tasks. The end goal here isn't just to send a few hardy microbes to Mars and call it a day. It's to create a thriving, interconnected system of bioengineered life that works together to make Mars habitable. Picture a Martian greenhouse filled with crops grown in soil enriched by engineered microbes, powered by biofuels produced just meters away, with oxygen generated on site for the humans tending to it all. Synthetic biology makes this vision possible, yet as exciting as this all sounds, it's not without challenges. How do you ensure these organisms behave as expected in such an alien environment? And how do you prevent unintended consequences like engineered microbes disrupting Mars' native conditions, if any exist? These are questions researchers are carefully considering as they push the boundaries of what synthetic biology can achieve. Still, the idea of designing life forms capable of reshaping Mars is more than just a scientific dream. It's becoming a tangible reality. With each breakthrough, we're moving closer to turning Mars from a barren wasteland into a second home for humanity. And through that same process, we're redefining what life itself can do, not only adapting to the universe's challenges, but actively transforming them to suit our needs. When it comes to living in space for the long haul, one of the most vital challenges is creating self-sufficient habitats environments that can sustain human life indefinitely without relying on constant shipments of resources from Earth. This is far from a simple task. On Earth, we live within a delicate balance maintained by countless interconnected processes. Plants produce oxygen, microbes break down waste, and water cycles endlessly through evaporation and precipitation. In space, however, none of these natural systems exist, and we're left with one pressing question: how can we recreate them in a completely artificial environment? The answer lies in the revolutionary potential of synthetic biology to design closed-loop bioregenerative systems, self-sustaining ecosystems capable of recycling waste into life-sustaining resources. At the heart of these systems is the concept of a closed loop. In a traditional space habitat, waste is a problem. It accumulates and must be either disposed of or stored, taking up precious space and resources. In a bioregenerative system, waste becomes an asset. Every byproduct, from carbon dioxide exhaled by humans to organic waste from plants, is fed back into the system to be converted into something useful. This is where engineered microbes step in as the unsung heroes. These tiny organisms are being specifically designed to perform essential recycling tasks, transforming what would otherwise be waste into vital resources like oxygen, clean water, and even food. Let's start with air. One of the most basic needs for human survival is oxygen, and synthetic biology offers elegant solutions to produce it in space. While traditional life support systems rely on mechanical air scrubbers to recycle carbon dioxide, these systems are energy-intensive and prone to failure over time. Instead, bioengineered organisms like algae or cyanobacteria can be introduced into the habitat. These organisms naturally consume carbon dioxide and release oxygen through photosynthesis. The twist: scientists are now modifying these microbes to maximize their efficiency in closed systems, making them ideal for producing oxygen in space habitats. Imagine an advanced bioreactor, compact yet capable of supplying breathable air for a colony, powered entirely by engineered life that continually renews itself. Next, let's talk water. In the void of space, every drop of water is precious. Engineered microbes can play a critical role in purifying and recycling water within a habitat. For instance, microbes can be programmed to break down organic waste in greywater, removing harmful contaminants and pathogens, and leaving behind clean, drinkable water. This process not only eliminates the need for bulky filtration systems, but also creates a sustainable loop where water is continuously reused, drastically reducing waste. But perhaps the most fascinating aspect of artificial ecosystems is how plants, our natural multitaskers, fit into the equation. Bioengineered plants could serve as more than just a food source in space. Imagine crops that are optimized to grow in low light conditions or require minimal water, while also performing critical environmental functions. These plants could be designed to filter air, removing impurities while releasing oxygen. Some could even purify water by absorbing and processing toxins through their roots, creating a natural filtration system. In this way, every plant in a space habitat could be a multifunctional powerhouse, contributing not just to nutrition but to the overall balance of the habitat's ecosystem. And then there's the possibility of engineering plants and microbes to work together in perfect harmony. For example, certain engineered bacteria could live symbiotically with plant roots, helping them absorb nutrients more efficiently in less than ideal conditions. This kind of partnership could allow plants to thrive in the microgravity of space or in artificial soil made from resources found on other planets, creating agricultural systems that are both sustainable and highly productive. The beauty of these artificial ecosystems is their adaptability. Once established, they can be fine-tuned to meet the specific needs of a habitat, whether it's a space station orbiting Earth or a Martian colony far from home. Engineers and scientists can adjust the genetic programming of organisms within the system to respond to new challenges, such as changes in population size or unforeseen environmental factors. This flexibility makes bioregenerative systems a powerful tool for ensuring the long-term survival of humanity in space. Ultimately, these systems aren't just about survival, they're about thriving. A habitat that can recycle its own air, water, and waste not only provides the resources humans need to live, but it also brings a sense of stability and independence. It's the difference between constantly relying on Earth as a lifeline and truly building a home in the stars. Synthetic biology is making this dream achievable, one engineered organism at a time. One of the biggest obstacles to building a sustainable human presence in space is the sheer cost and complexity of transporting everything we need from Earth. Every kilogram sent into orbit requires vast amounts of energy and resources, and the further we travel, to the Moon, Mars, or beyond, the more unsustainable this becomes. Take building materials, for example. Sending construction supplies to Mars for a colony would add billions of dollars to the mission's price tag. But what if we didn't have to bring everything from Earth? What if we could use the resources already present in space, combined with the power of synthetic biology, to produce the materials we need on site? This is the promise of biomanufacturing. Biomanufacturing is a cutting-edge approach to production that uses engineered microbes to produce a wide range of materials, from plastics to pharmaceuticals, using simple inputs. And in the context of space, it's a game changer. Instead of launching pre-made supplies, we could send compact bioreactors loaded with microbes tailored to make exactly what we need, on demand and using the raw resources of the Moon, Mars, or even asteroids. This process is called In Situ Resource Utilization, or ISRU, and it's poised to transform the way we think about space colonization. Here's how it works. Scientists can engineer microbes like bacteria or yeast to use local resources as their feedstock. For example, the carbon dioxide abundant in Mars's atmosphere could be captured and combined with water extracted from Martian ice to create sugars. These sugars, in turn, can fuel microbes that produce a variety of essential materials. Take bioplastics, for instance. By engineering bacteria to convert carbon and hydrogen into polymers, we could produce lightweight, durable materials for constructing everything from tools to habitat components directly on site. These plastics could even be used as feedstock for 3D printers, enabling astronauts to create custom parts and equipment as needed. The potential doesn't stop there. Synthetic biology is also unlocking new ways to produce medicines and other critical supplies. On long-duration space missions, carrying every possible drug for every potential scenario is impractical. But with biomanufacturing, we don't have to. Instead, microbes could be engineered to produce pharmaceuticals on demand, using only a small selection of precursor molecules. Need an antibiotic or painkiller? A bioreactor loaded with the right microbes could produce it in hours. This capability could not only reduce mission payloads but also ensure astronauts have access to life-saving treatments, even years into a mission. And then there's the possibility of producing building materials. Transporting construction supplies to another planet is wildly expensive, but microbes could give us a scalable, sustainable alternative. Some researchers are exploring how bacteria could be engineered to produce bioconcrete using Martian or lunar regolith as a starting material. Others are looking into the potential of growing mycelium, the root-like structures of fungi, to create lightweight, durable structures that can be used as part of habitat walls or insulation. By using local materials and biological processes, we could drastically reduce the need for heavy, prefabricated construction elements and instead grow what we need where we need it. Even metals could be within reach. Certain microbes already found on Earth are capable of extracting valuable metals from rocks, a process known as bioleaching. By modifying these microbes for use on the Moon or Mars, we could potentially extract iron, nickel, or other critical elements from the local geology. These materials could then be used for building essential infrastructure from solar panels to vehicles and eliminate the need for extensive mining operations. One of the most exciting aspects of biomanufacturing is its scalability and adaptability. The same microbes can often be re-engineered to produce entirely different products depending on the mission's needs. This flexibility is crucial for space exploration, where circumstances can change quickly and redundancy is critical. Instead of carrying large stockpiles of spare parts and supplies, future space missions could bring the genetic instructions to program microbes on the fly, essentially printing what's needed from biological blueprints. The implications of biomanufacturing are huge. It enables true independence from Earth, allowing future space colonies to rely on locally sourced, renewable resources instead of costly and finite shipments. More importantly, it's a sustainable approach that aligns with the long-term goal of creating self-sufficient human settlements in space. Synthetic biology is redefining the rules of resource management, production, and sustainability. It's not just about surviving in space, it's about thriving, creating a system where life supports life, no matter how far we travel from Earth. And with biomanufacturing, the challenges of building in the cosmos become opportunities to innovate at the frontier of human ingenuity. With the power to rewrite life itself, synthetic biology offers incredible possibilities. But it also comes with significant ethical dilemmas and risks that we cannot underestimate, especially when applied to space exploration. The act of engineering organisms to survive in harsh, alien environments is an unprecedented leap for science, but it also raises profound questions about responsibility, unintended consequences, and security. As we venture into this frontier, it's crucial that we address the ethical considerations and potential risks head-on. one of the most pressing concerns revolves around the unintended ecological impact of releasing bioengineered organisms into extraterrestrial environments. For now, we assume that places like Mars and the Moon are sterile, void of any existing life. But what if we're wrong? Introducing genetically modified organisms, from microbes to plants, could fundamentally alter any undiscovered native ecosystems, even on a microbial level. These engineered life forms could outcompete or even wipe out potential indigenous organisms before we ever have the chance to study them. In this way, synthetic biology might inadvertently destroy what it sought to explore, any clues about alien life or the natural processes of those planets. And even if no other life exists, the changes we make to these environments are irreversible. Engineering Mars to suit human needs, for example, could forever erase its original state, posing a philosophical question. Do humans have the right to terraform or permanently alter other worlds? On Earth, we've seen numerous examples of the unintended consequences of introducing foreign species into new ecosystems. Cane toads in Australia or invasive algae in water systems across the globe have caused significant environmental damage, often spiraling far beyond human control. In space, where the stakes are even higher, we must ensure we don't make the same mistakes. The release of bioengineered organisms into other worlds needs to be carefully regulated and studied, ideally replicating their behavior in controlled environments before ever taking them off Earth. Beyond ecological impacts, there's also the risk of misuse. Synthetic biology has tremendous potential for good, but in the wrong hands, it could lead to catastrophic outcomes. Genetic engineering tools like CRISPR are becoming more accessible, and while this democratization of technology is exciting, it also raises fears of bioterrorism. In space, where humans operate in fragile, enclosed environments, the release of a harmful bioengineered organism, either unintentionally or maliciously, could have devastating effects. A single engineered pathogen could compromise an entire space station or colony, as well as the systems designed to sustain human life. The risks are compounded by the unique conditions of space, such as microgravity and limited access to medical care, which could make containing or responding to such threats even more challenging. There's also the ethical question of how far we should go in augmenting organisms, including ourselves, to adapt to space. Should we engineer plants and microbes to survive on Mars, or should we instead adapt human biology to endure those challenges? Gene editing technologies have already sparked debate about altering human DNA to prevent diseases. Extending this to create humans capable of tolerating space radiation or low gravity enters ethically murky territory. While these modifications could enhance survival, they blur the lines between natural evolution and deliberate design, raising concerns about consent, equity, and the potential for misuse. Given these risks, the need for strong regulations and oversight in synthetic biology cannot be overstated. Scientists, policymakers, and ethicists must work together to establish international guidelines that govern the use of these technologies, particularly in space. These regulations should address everything from the safe containment and testing of bioengineered organisms to the ethical boundaries of genetic modifications to stringent protocols for preventing bioterrorism. Collaboration on a global scale will be essential, as the consequences of any misstep won't just affect one nation, they could impact humanity as a whole. Public transparency is another critical element. The more informed people are about the risks and benefits of synthetic biology, the better equipped society will be to make decisions about its use. Building public trust requires openness about the intentions behind these technologies, as well as a willingness to engage with diverse perspectives on what is ethical and acceptable. The potential of synthetic biology to help humanity explore and inhabit space is extraordinary, but with this potential comes a weighty moral responsibility. As we shape life to suit our needs, we must ensure we're also safeguarding ecosystems, protecting human life, and respecting the unknown. The choices we make today will define not only how we explore the universe, but how we view ourselves as stewards of life on Earth and beyond. Synthetic biology isn't just a futuristic concept, it's already being tested and applied in some of the most ambitious space missions happening right now. Across both public and private sectors, scientists and innovators are laying the groundwork for how bioengineered organisms can support life beyond Earth. Let's explore some real-world examples that showcase synthetic biology in action and highlight its growing importance in the quest for space exploration. One of the most prominent players in this field is NASA, which has been at the forefront of experimenting with bioengineered microbes aboard the International Space Station. The microgravity environment of the station presents a unique opportunity to study how these organisms behave and function in space. For instance, NASA has been investigating how engineered microbes can be used to convert astronaut waste into valuable resources like food, nutrients, and even bioplastics. This type of recycling system could one day be integral to long-term missions, turning what would otherwise be a waste problem into a self-sustaining cycle of resource production. Another groundbreaking NASA initiative involves developing microbes capable of mining metals. This process, known as biomining, uses bacteria to extract valuable metals like iron and rare earth elements from rocks. On the International Space Station, experiments have already been conducted to see whether these microbes can perform just as effectively in microgravity as they do on Earth. If successful, biomining could one day be used on the Moon, Mars, or even asteroids to harvest essential materials for construction and technology, eliminating the need to transport such resources from Earth. But NASA isn't the only organization exploring synthetic biology in space. The private sector is also playing a key role, with companies like SpaceX and Blue Origin incorporating advanced biotechnologies into their strategies for space colonization. SpaceX, for example, has long envisioned Mars colonization as humanity's next great leap. While synthetic biology isn't their primary focus, SpaceX's ambitions have spurred collaborations with researchers and biotech companies exploring ways to terraform Mars or create self-sustaining life support systems. The vision is clear. Bioengineered organisms could help convert Martian resources into breathable air, drinkable water, and even building materials. Meanwhile, Blue Origin, led by Jeff Bezos, has expressed a strong interest in creating sustainable human settlements in space. The company's vision of moving heavy industries to space aligns closely with the goals of synthetic biology. For example, future Blue Origin missions could incorporate biomanufacturing techniques to produce lightweight materials in orbit or on extraterrestrial surfaces, drastically reducing the cost and complexity of transporting goods from Earth. Synthetic biology isn't limited to the biggest names in aerospace either. Smaller biotech-focused companies and research institutions are rapidly advancing the field. For instance, a company called Made in Space has already demonstrated how microorganisms can be used in microgravity to manufacture materials like Z-blan, a specialized type of optical fiber. While these experiments are in their early stages, they showcase how bioengineered organisms could one day help produce high-value materials and supplies for space missions. One particularly exciting example is the work being done by the European Space Agency. ESA is exploring how modified cyanobacteria might be used to support human life on Mars. These microbes are being designed to produce oxygen and consume carbon dioxide, mimicking the role of plants in Earth's ecosystems. The goal is to create a scalable system that can eventually support a colony, providing breathable air and even components for biofuels. Even beyond Earth's orbit, synthetic biology is expanding its potential applications. Researchers are already considering how bioengineered organisms could play a role in deep space missions, like those to Jupiter's moon Europa or Saturn's moon Titan. These missions would require systems capable of functioning autonomously for years, and synthetic biology offers a way to ensure the sustainability of such expeditions. Microbes could be deployed to process local resources for food, energy, or oxygen, enabling humans to explore far deeper into the cosmos than current technologies allow. These case studies represent just the beginning of what synthetic biology can achieve in space exploration. Through a combination of public and private sector innovation, this technology is steadily moving from the lab to real-world applications, with every experiment helping us to refine and expand its potential. By leveraging bioengineered organisms, we're not just making space exploration more feasible, we're setting the stage to truly thrive beyond Earth. As we look ahead, the intersection of synthetic biology and space exploration promises to unlock possibilities that once belonged solely to the realm of science fiction. The future isn't just about tweaking existing organisms to suit space environments, it's about reimagining life itself to thrive where nature never intended. From bioengineered humans capable of enduring the extreme conditions of space to entirely synthetic organisms designed for specific extraterrestrial tasks, the coming decades could redefine humanity's relationship with biology, technology, and the universe. One of the boldest ideas on the horizon is the concept of engineering humans to better adapt to space. Space is notoriously hostile to the human body. Microgravity weakens bones and muscles, while cosmic radiation poses serious risks to DNA, leading to increased chances of cancer and other health issues. Instead of relying entirely on protective equipment and shielding, synthetic biology offers a radically different approach, modifying the human genome itself. Scientists are already exploring genes found in extremophiles organisms that live in Earth's harshest conditions, such as deep-sea vents or radioactive environments, to see if those traits could be applied to human biology. Imagine humans with enhanced DNA repair mechanisms to resist radiation damage, or with physiological adaptations to minimize the effects of long-term weightlessness. These modifications could fundamentally change how we approach long-term space travel and colonization, making it possible for humans to not just survive, but truly live in environments like Mars or even deep space. However, bioengineering human traits comes with its own ethical challenges. Who decides which enhancements are acceptable? How do we balance the risks of genetic modification with its potential benefits? And what happens when we start blurring the line between what it means to be human and what it means to be engineered? These questions will need to be tackled as the frontier of synthetic biology advances, but the possibilities are too significant to dismiss. Equally exciting is the creation of entirely synthetic organisms, life forms that don't evolve naturally but are designed and built from scratch in a lab. Using tools like CRISPR and advanced gene writing technologies, scientists could design microbes with customized capabilities for specific extraterrestrial environments. For instance, a synthetic organism could be constructed to extract rare gases from Martian soil or metabolize methane present in the atmosphere of Saturn's moon Titan to generate energy. On icy moons like Europa, where vast oceans lie beneath thick crusts of ice, completely synthetic microbes could be engineered to explore these alien waters, searching for resources or even unlocking secrets about the origins of life itself. The beauty of designing synthetic organisms is their potential to go beyond Earth's biological limitations. Traditional bioengineered organisms, like modified plants or microbes, are often constrained by their evolutionary history. They can only be modified so much before they stop functioning effectively. Synthetic organisms, by contrast, are a blank slate. Scientists can build them from the ground up, ensuring they're perfectly suited for their intended tasks. In the future, these creatures could serve as the foundation of self-sustaining ecosystems on other planets, converting raw materials into food, air, or fuel. Advancements in artificial intelligence and machine learning are set to supercharge this process. Designing complex genetic systems is no easy task. Current methods involve trial and error, requiring meticulous testing to ensure organisms behave as expected. AI, however, can simulate millions, if not billions, of genetic variations in silico or in a computer environment far faster than any human researcher. This allows scientists to predict which genetic designs are most likely to succeed before they are ever created in the lab. In essence, AI acts as a guide, helping to navigate the vast genetic landscape and accelerate the pace of discovery. Moreover, machine learning algorithms could assist in monitoring and optimizing synthetic ecosystems in real time. Consider a Martian colony with bioengineered plants, microbes, and other organisms working together to recycle air, water, and waste. If one component of the ecosystem begins to malfunction, say, a drop in oxygen levels or a rise in harmful bacteria, AI systems could immediately detect the issue, analyze the data, and suggest adjustments to restore equilibrium. This type of dynamic, AI-driven management will be critical as we rely more heavily on bioregenerative life support systems in space. Looking even further into the future, synthetic biology may allow us to move beyond adapting life to existing planetary conditions and instead create entirely new biospheres tailored to human needs. Imagine terraforming a barren exoplanet by introducing a suite of synthetic organisms designed to work together: microbes that enrich the soil, plants that produce oxygen, and fungi that stabilize the environment. Over time, this artificially created ecosystem could transform the planet into a livable space, effectively turning a dead world into humanity's next home. While these ideas might seem like far-off dreams, the pace of innovation in synthetic biology, AI, and space exploration suggests that they may be closer than we think. As these technologies continue to converge, the possibilities for designing life in space are truly limitless. But with this power comes responsibility. Every step forward will require rigorous oversight, ethical deliberation, and a commitment to using biology not only to advance humanity's ambitions, but to do so in ways that respect and preserve the universe's natural wonders. The future of artificial life in space is not just about science or technology, it's about redefining what it means to exist in the cosmos. And as we take these monumental steps, we're not just shaping the future of space exploration, we're reshaping the very fabric of life itself. Synthetic biology stands at the crossroads of innovation and the cosmic frontier, offering humanity an unprecedented opportunity to redefine life as we know it. By crafting artificial life designed to overcome extraterrestrial challenges, we are no longer confined by the limitations of Earth's biology. Instead, we can engineer solutions to create breathable air, grow food in barren soils, produce essential materials, and even protect ourselves from the relentless dangers of space. This revolutionary technology transforms the seemingly impossible task of space colonization into a tangible, achievable goal. Think about what we've explored in this journey: microbes engineered to terraform Martian soil, organisms designed to recycle waste into valuable resources, and the potential to produce medicines, fuel, and oxygen on demand in the most hostile environments imaginable. These aren't just hypothetical ideas, they are real technologies being developed and tested today. With synthetic biology, we're not merely visiting other worlds, we're giving ourselves the means to build sustainable homes there. Whether it's creating closed-loop ecosystems that allow astronauts to survive indefinitely or engineering entire biospheres capable of supporting new civilizations, synthetic biology is at the heart of a bold new vision for humanity's future among the stars. But this isn't just about survival, it's about thriving. By harnessing the principles of biology and innovation, we stand on the edge of transforming barren landscapes into vibrant, life-supporting environments. The questions we face now are no longer about if we can do this, but about how we can do it responsibly. How do we ensure that the technology we create serves humanity without unintended consequences? And how do we balance scientific progress with the ethical stewardship of our newfound power to shape life itself? These are the challenges we must address as we move closer to making interplanetary civilization a reality. What's clear is that synthetic biology is not just another tool for space exploration. It's the key to unlocking humanity's potential to become an interplanetary species. This isn't just about conquering new frontiers, it's about growing as a species, expanding our understanding of life, and redefining what it means to call a planet home. The possibilities are as infinite as the cosmos itself. So, what do you think? Are we ready to take this monumental step and create life designed for the stars? Does the idea of reshaping biology to conquer new worlds excite you? Or does it raise questions about how far we should go? Let us know your thoughts in the comments below. We'd love to hear what you think about this fascinating frontier. If this video inspired you to think differently about humanity's future, give it a thumbs up and share it with others who might be curious about the possibilities ahead. And don't forget to subscribe for more content exploring the incredible breakthroughs shaping tomorrow. Together, we can explore what's next for humanity and beyond.